Okay, our next chapter is going to deal with infectious disease, how it's produced, how it spreads. Okay, we'll talk about the relationships between host, microbiome, and pathogen, talk about concepts of infection and infectious disease, and talk about how disease impacts communities and how communities shape emerging pathogens. In this section, we'll talk about the difference between the microbiota and the pathogenic microbiota. We'll talk about the relationship between infection and disease and between virulence and pathogenicity, differentiate between infectious dose and lethal dose, and talk about the fundamental attributes of a pathogen. Okay? A lot of areas of the human body are colonized with microbes. Like I told you earlier, you don't live in a sterile world, right? You have stuff growing on you, in you, and around you all the time, right? The relationship between the human host and their microbiota include mutualism, commensalism, and parasitism. Okay, um, a parasite is a microbe that harms the host and exclusively benefits. Okay, um, examples would include ectoparasites that live on the body's surface and endoparasites that live inside the body. Okay, now parasites tend to be multicellular. Okay, but there are exceptions to that. Okay, pathogens are bacterial parasites of humans. Okay, um, and pathogenicity is the ability of that particular organism to cause disease. Okay, it depends on the genetics of the host and the virulence factors of the pathogen. Now, what are virulence factors? They're traits of the pathogen that allow it to make the disease. Okay, for instance, the pathogen could produce a toxin on its surface or secrete a toxin. Okay, that causes disease. Um, the pathogen could suppress the immune response designed to destroy it. Okay, the pathogen could um, produce uh, decoy antigens in order to send the immune system on a wild goose chase so that it can't find and destroy the pathogen. Those are just examples of virulence factors. There's all kinds of different virulence factors, okay? And each pathogen has a unique weapon that it can use in order to cause illness, disease, okay? So the virulence describes the level of harm caused by a pathogen following the infection. Virulence is determined by factors that can promote attachment and colonization of the tissue, okay, meaning the ability to hook up and grow within the tissue. We call that invasion. Factors that can facilitate pathogen spread from the initial site of the infection, which we call invasiveness, or have a toxic effect on cells, tissues, and host physiology. We call those toxins, right? Toxins can be secreted, called exotoxins, or they can be on the surface of the pathogen, where they're called endotoxins. Primary pathogens are likely to cause disease after an infection in a healthy host. They rapidly reproduce and increase in number. They have moderate to high virulence. Okay. Opportunistic pathogens are only going to hit you if your body's defenses are compromised, meaning there's pre-existing disease, or you're undergoing a treatment that may suppress your immune system, or maybe you're extremely old or extremely young. Okay, all of those would be targets for opportunistic pathogens. Examples: Shigella flexneri causes bacillary dysentery, and when ingested, it can survive the natural barrier of the acidic stomach, enter the intestine, and begin to reproduce. Okay, Treponema pallidum causes syphilis because it can survive the initial attack by a healthy immune system and spread through the body and the blood. Okay, another example: Helicobacter pylori can colonize the lining of the stomach because it has um, surface molecules, okay, that can latch on to the stomach lining, and it produces an enzyme called urease that neutralizes stomach acid while it feeds off of the food that drops into the stomach from the esophagus. Okay? LD50 stands for lethal dose 50%. Okay? Each LD50 measurement requires infecting small groups of animals with increasing numbers of infectious agent and observing how many animals die. The number of microbes that kill half the animals is going to be the LD50. In this example, Agent 1 requires fewer organisms to kill half the animals than Agent 2. Agent 1 is thus more virulent. Okay? Most LD50s are the result of information that we gather from organisms other than humans, although there are some human LD50 data that we can use as well. Okay? As a result of things like accidental exposure. Basically, the more lethal something is, right, the smaller the LD50 is going to be. Okay, an example, right, the LD50 for cyanide is much lower than the LD50 for water. Okay, but you can die by ingesting either one. But the amount of cyanide you'd have to take in would be much less than the amount of water you would have to take in. Okay? So everything on the planet has an LD50. Okay? The host range is basically what the targets are for the organism, right? Some organisms can infect only one type of host to make disease. The example of a narrow host range would be Salmonella enterica, which has a serovir that infects humans only. Okay, that's serovir means serological variant. Okay, broad host range, Salmonella enterica with the serovir typhonurium infects other animals in addition to humans. Okay, so you've got two different flavors of the same organism. Okay, distinguished by the antigens on their surface. Okay. Treponema palladium is another example of a narrow host range that only is going to hit humans. Okay. A serovar is just defined as a subclass within a species. We'll talk about the signs and symptoms of disease and talk about the role of immunopathogenesis in infectious disease and talk about the 5 basic stages of infectious disease. Okay? A disease literally means what it says. It means not at ease. Okay? Dis meaning missing or absent, and ease meaning at ease. Okay? An infectious disease is one that's caused by a microbe, and it can be transferred from one host to another. And the method of transfer is going to vary depending on the microbe. Okay? Some are airborne. Some are spread by contact only with things like infected bodily fluids. Okay? It all depends on what type of pathogen we're talking about. Okay? An acute disease is one that comes on suddenly. An example would be strep throat. A chronic disease takes place over a longer period of time. An example would be tuberculosis or TB. The subacute disease is something like endocarditis, which is an inflammation of the endocardium, which is the inner lining of the heart. Okay, and then latent disease would be example would be herpes simplex, which initially manifests as chickenpox, but in adults can reemerge as something called shingles. Okay, and that is a viral disease. Okay? Diseases are identified by their signs and symptoms, right? A sign is something that we can put a number on. Okay, we observe them by examination. Things like a particular body temperature or a particular blood pressure, right? The presence or absence of a rash. Okay, symptoms are experienced by the person, and they are subjective indications of disease. Examples would be things like pain and tiredness, also known as fatigue. Sequelae are pathological consequences that occur after the infectious disease is over. Okay, an example is strep throat resulting in heart or kidney damage. 2 sequelae known as rheumatic fever and glomerulonephritis account for this, right? So while the strep throat's going on, all you're concerned with is the pain, right? And then afterwards, later, you end up with organ damage. Some sequelae can actually be caused by the host's response to the infection in an attempt to destroy the pathogen. You end up destroying healthy tissue. An example would be the spirochete that causes Lyme disease, which is brought on by a vector known as the tick. Right? The tick takes a blood meal, he bites you, he injects you with the spirochete. As a result, the spirochete basically works its way through tissues and tags healthy cells with antigen that are part of the surface of the spirochete, but the spirochete leaves the area, and the result is that your immune system then moves in thinking those cells are infected when in fact they've only been hit with an antigen dose and destroys them. Okay, and this results in some cases autoimmune problems long after the bacteria, the spirochete's been wiped out. Okay, a syndrome is a group of symptoms that consistently occur together and collectively characterize a particular condition. An example would be chronic fatigue syndrome, where we see extreme fatigue at around 6 months and then worsening fatigue after physical and mental activity. You're still tired after you sleep. You have orthostatic intolerance, meaning it's difficult for you to stand up, okay? And you have problems with thinking and memory, okay? These are all clustered together, okay? Chronic fatigue, in some cases, is thought to be brought about by a virus known as Epstein-Barr, okay? You can see here a chart showing you some acute infectious diseases, and the brown bar shows you the number of microbes, and the blue bar is the immune response, right? After initial infection, the number of microbes rise in the body dramatically until the immune system recognizes that the invader is present, and then eventually comes up with an immune response. We call this our adaptive immunity, right? This portion is known as the prodromal stage. The symptoms develop at the same time that the immune system begins attacking the organism of the virus. During the illness phase, the pathogen reaches its peak numbers, and then once the immune system is effective at knocking the pathogen down, we enter the decline phase. The immune system remains ready during convalescence, but the ability to attack the same invader will eventually decline over time. Okay? However, we should point out that during the course of fighting this infection, in the event that there is a second infection of the exact same variant, right, exact same strain of bacteria or type of virus, right, that the immune response will be more effective than the initial response to that infection. Okay, as we age, all our body systems decline in function because the rate of cell death outstrips the rate of cell replacement, and so that includes our immune response. Okay. Public health agencies track disease rates and severity. Two stats that are kept are morbidity and mortality. Morbidity is the rate of illness due to a disease, while mortality is the rate of death due to a disease. The CDC publishes the Morbidity and Mortality Weekly Report, and it discusses current outbreaks, stats, and health topics that affect people in the U.S. We'll talk about some complex versus simple infection cycles and endemic, epidemic, and pandemic disease, and talk about some animal reservoirs that sometimes serve as incubators for disease. Okay? An example here is malaria, right? Infectious diseases can be transmitted horizontally from one member of a species to another by different means, right? Direct contact between people, indirect transmission by fomites or insect vectors. Now, you're going to say, what the heck is a fomite? A fomite could be an object, okay, such as a door handle or a tissue, Okay, a vector, an insect vector, is simply an insect that harbors the pathogen on it or in it and spreads it by feeding or landing or burrowing in the host, right? You can also have vehicle transmission by air, food, and water, okay? And vertical transmission from parent to offspring during birth, such as passing through an infected birth canal, okay? All of these are possibilities. What you're looking at here is just an example of the malaria life cycle, right? Um, Basically, there are parts of the world where malaria is what's called an endemic disease, meaning it's always in the area. Okay, um, if we look at malaria, okay, microbes can be transmitted by direct contact or by aerosols. Right, you can sneeze, and if you don't cover your mouth, right, you can spread infectious particles directly from an infected to an uninfected individual. Okay, um, this would be, for instance, like the flu, or right now we're undergoing the precautions due to coronavirus. Okay, in malaria, the insect vector is a major player. Okay, what happens is that the Anopheles mosquito harbors the agent of malaria, which is a protozoan, right? The insect takes a blood meal from the host, right? And as a result, sends the pathogen into the host, okay? The result is that the pathogen reproduces, all right? And then when an uninfected mosquito takes a blood meal, right, then they pick up the malarial parasite, and then they can end up spreading it to an uninfected individual. Okay, so that's the malaria life cycle, right? You got this what they call vertical transmission. Okay, insect vectors can also be agents of accidental transmission, such as biting an infected bird with West Nile and biting a human being. Okay, this is less common than incidences like this direct transmission or this natural transmission through the insect vector. Okay, pathogens can be transmitted both directly and indirectly through contact with a reservoir. Right, uh, for instance, cats can carry certain viral infections or um, parasitic infections that can be transmitted to humans. Okay. Um, then there's vehicle transmission, right? You can have contaminated food or a fomite, like I said, a door handle or a napkin or a tissue. Okay, the infected person touches it, then you touch it, and then now you've got it. Okay, so these are all examples of ways that the infection can move around. Right. Cycles of infection can be simple or complex, right? Direct contact, person to person, right? Touching, kissing, intercourse, right? Aerosols, okay? Rhinovirus can be passed from person to person by aerosols of secretion particles called droplet nuclei, which hang in the air for up to 15 minutes after a sneeze, okay? That's one of the reasons we get colds more often in the wintertime than when it's warm out, because we're inside more. More people sneeze, more people come into contact with the droplets, and thus they get sick. Okay. Indirect transmission is where the pathogen can spread indirectly through an intermediary, which may be either living or non-living. A fomite would be an inanimate object, right? Or you could have a vehicle, such as food, water, or air. Okay. Or you can have an insect vector, such as a tick or a mosquito, which would spread the pathogen in a way it wouldn't normally spread if it didn't have a vector. Okay? Examples, right? Shigella can be passed directly or indirectly through inanimate objects called fomites, such as a shared fork. This is also known as vehicle transmission, right? Rickettsia is passed through a tick vector. This is the agent of yellow fever, West Nile, and malaria, which also pass through insect vectors. In that case, passes through the mosquito, right? The mosquito harbors the pathogen in its gut. It takes a blood meal, and then it gives you a dose of the pathogen. Okay. A reservoir is an animal, which can include humans, or an environment that normally harbors the pathogen. Okay. Examples, right? Rocky Mountain spotted fever is a tick reservoir, and the vector is Rickettsia rickettsii. Right? Lyme disease also tick spread, as well as mice and small mammals, right? Carrying the ticks. Okay, so the ticks are the vector, and the mice also aid in the transmission because they serve as the reservoir, right? The tick can bite the mouse, and then the tick can bite you, right? An example would be Borrelia burgdorferi, right? Syphilis, where humans are the reservoir, and transmission of the pathogen Treponema pallidum is through direct contact with the reservoir, right? Reservoirs are crucial for the survival of a pathogen and serve as a source of the infection. They don't necessarily exhibit any disease, whereas a carrier is a special type of reservoir. Examples, right? Neisseria meningitidis is maintained in a population by infecting the nasopharynx of unsuspecting hosts whose immune system keeps the bacteria out of the blood. A simple sneeze from a carrier can aerosolize the pathogen and transfer it to a susceptible host. Okay? So let's take a look at cycles of disease transmission.
Thanks for stopping by. This is 2 Minute Classroom, and today we're talking about how epidemiologists classify the spread or prevalence of health issues such as a disease or virus. We'll talk specifically about endemic, epidemic, and pandemic diseases. An endemic health issue is a disease, virus, or other health concern that is constantly prevalent in a given location or population. The number of affected individuals is generally low and does not significantly increase or decrease over time. It's just always there. Examples include chickenpox, which affects children at a regular, predictable rate, and malaria, which is constantly present in many parts of Africa. You might think something like the flu is an endemic virus, but it is, in most geographies, it behaves more like our next classification, epidemics. An epidemic health issue is one that has a sudden and rapid rise in a particular region or population. The seasonal flu behaves more like an epidemic because cases spike at certain times of the year. Other epidemics throughout history include yellow fever, which killed 5,000 people in Philadelphia in 1793, and cholera, which hit the U.S. in 3 waves, killing over 150,000 Americans, and is still very prevalent and fatal in many parts of the world. An epidemic becomes a pandemic when it spreads across the globe. Essentially, an epidemic is not limited to a specific geography or population. It also usually affects a significant number of people. Some of the most prolific pandemics throughout history include the HIV/AIDS pandemic, which has claimed the lives of more than 36 million people since 1981, and with over 30 million cases still prevalent throughout the world. The Spanish flu in 1918, which infected approximately 500 million people and had an estimated mortality rate of 10 to 20%, and the bubonic plague, which spread across Europe, Asia, and Africa, killing an estimated 75 to 200 million people. Now for some positivity. Modern medicine and hygiene have come a long way. We have many vaccines that protect us from everything from the flu to polio. We know much more about how to prepare food so that it is safe to eat and free from bacteria, and we have much better understanding of how diseases spread so that we can react quickly to stop the progression of many potential epidemics and pandemics. Now watch these videos that you may find interesting, or check out my sources in the description to learn more about the topics discussed in this video. Thanks for watching, and I'll catch you next time.
Okay, a few other concepts we need to be familiar with. Zoonotic diseases. Where have you seen the word zoo before, right? You go to the zoo to see animals, right? These are diseases that are spread by infected animals, and they can be transmitted to humans. The pathogen may or may not cause the animal reservoir to have a disease. So the animal may or may not be sick. Okay? Transmission may be direct or indirect. So you can have spillovers from animal to human. An example would be the virus that can be spread from pigs to humans, commonly known as H1N1. Okay, Lyme disease, right? The pathogen doesn't cause disease in the animal host, which is the tick, but it causes the disease when the bacteria are introduced into the human bloodstream when the tick takes a blood meal. Ebola is a virus that causes disease in the animal host and also in the human. Okay, the problem, of course, with Ebola from a strategy standpoint is that it kills the host relatively quickly. Okay, the mortality rate is extremely high in Ebola, and that limits its ability to spread. Okay. We'll talk about portals of entry and exit for pathogens and discuss biosafety and containment. So how do you get infected? Okay. Well, any part of the body that's exposed to the outside world has the potential for infection. Okay. And that's one of the reasons why in the human, right, the respiratory, the digestive, the urinary, and reproductive systems are all portals of entry and exit. Okay. As are other openings in the body, such as the orbits that hold the eyes and the ear canals. And there are even some routes that can lead back to the brain in certain instances. Okay, for instance, the olfactory bulb, which sits on top of the nasal cavity, all right, can sometimes be utilized by certain pathogens to work their way back into the brain. This is how people contract the brain-eating amoeba, by swimming in contaminated water. Okay, but normally you have to have an opening, right? And that's what allows the infection to take hold. Okay, fecal oral route, where the portal is the mucosa of the GI tract. Remember that the mucosa is simply the inner lining of any body system that's open to the outside world, and it's characterized by the fact that it secretes mucus, which normally traps and clears pathogens. Okay, skin, the portal on the skin is the epithelial tissue. Respiratory, where the portal is the mucosa of the respiratory tract. Urogenital, the mucosa of the genital and urinary tracts. And parenteral, where an injury is the port of entry. Okay, could be a break in the skin, could be a break in the mucosa. Okay, and then we can tunnel our way through from there. All right. Next thing we want to talk about are biosafety levels. Okay, so let's take a listen.
Biosafety levels are a set of biocontainment controls that are required to separate the biological agents based on the risk they cause on the environment and human beings. These levels are ranked from 1 to 4. Each level has specific controls for containment of biological agents and microbes. The controls are based on the infectivity of the disease, severity of the disease, source of the agent, and route of invasion into human body. These biosafety levels play an important role in designing the facility, safety environment, and laboratory practices. Each biosafety level has its own standard laboratory practices that one must follow. However, each microbiology laboratory, irrespective of their biosafety level, must follow the basic good laboratory practices. Let us discuss about each biosafety level individually. Biosafety Level 1, or BSL-1. As the lowest biosafety level, it includes the microbes that are non-pathogenic. Laboratory personnel can work with minimum risk in these facilities. BSL-1 labs do not need special containment equipment. People can work on open bench tops. These labs are typically used for students and trainee microbiologists. Example of BSL-1 organism will include the non-pathogenic strain of E. coli. However, basic safety practices must be followed in these facilities. Biosafety Level 2. The Biosafety Level 2 is for microbes that are associated with human diseases. That means, pathogenic or infectious bacteria and viruses are included in this level. Examples of BSL-2 microbes may include Staphylococcus and hepatitis virus. BSL-2 labs are expected to have more controls than BSL-1. In addition to the standard laboratory practices of BSL-1, these practices must be followed in BSL-2 labs. Biosafety Level 3. This level includes the microbes or agents that can cause serious or potentially fatal diseases through inhalation. BSL-3 facilities are usually under the control of government agencies, and the laboratory staff is under medical surveillance. Examples of BSL-3 microbes may include yellow fever and the bacteria that cause tuberculosis. BSL-3 facilities should maintain unidirectional airflow from clean air to the infectious air. The recirculation of air happens through HEPA filters. Additional laboratory practices must include the following. Biosafety Level 4. The BSL-4 facilities are rare in the world, being the highest level of biological safety. The BSL-4 microbes cause fatal infections. The diseases caused by these agents usually do not have vaccines or treatment. Two examples of such microbes include Ebola and Marburg viruses. The BSL-4 facility must be isolated and have dedicated supply air and exhaust air. In addition to the practices of other BSL laboratories, these practices must be followed in BSL-4 labs. If you liked the video, please hit the like button or comment and share. Please do subscribe to my channel and press the bell icon to get notified on the upcoming videos.
So some examples of each of these. Group 1, Bacillus subtilis, or E. coli strain K12, or Saccharomyces species. That would be BSL-1. Group 2, Bordetella pertussis, Clostridium species, Helicobacter pylori, Staph aureus. And then Group 3, Bacillus anthracis, Francisella tularensis, and Mycobacterium tuberculosis. And then finally Group 4, the most serious group, Ebola virus, hantavirus, Marburg virus. Okay, so just a few examples. We'll talk about the biological features of human hosts that influence the course of an infection, and talk about host behavior impacting our susceptibility to disease. Okay, so we would call these risk factors, right? In other words, if you're in this group, you're more likely to get infected. Okay, one of these is age, right? The greatest susceptibility is among the very young and the very old. Now, why is this? Primarily because the immune system is less effective in these age groups. Okay, in those under 3, the immune system has yet to fully form, and in those over 60, what's happened is that the cellular components of the immune system are dying more rapidly than they're being replaced. Okay, genetic makeup, right? You have to have the right kind of cell surface for the bacteria or the virus to dock and then infect, right? For something to colonize you, it has to cling to you, right? And not everybody is going to inherit a clingable surface, okay? In every case, for every pathogen. Behavior, right? One of the easiest ways to break the chain of infection is through hand washing, okay? Proper cooking of food, another critical component. Basically safe sex, right? So that you don't transmit STDs, sexually transmitted diseases, which are both viral, fungal, and bacterial in origin. Nutrition and exercise, right? The better your exercise and nutrition regimen, the more effective your immune system is going to be. And we could add to this stress levels, right? The more stressed you are, the less effective your immune system becomes, and that is thought to be linked to cortisol levels. And cortisol, among other things, has a role in controlling inflammation, which is an important process to rid us of disease. Okay. Other factors, right? The genetics. There are variants in the immune system, genes that can influence the course of an infection. Some people are going to be more, and some are going to be less susceptible to even an initial infection because of the components of their immune system. Okay. Remember that all our immune systems are functionally identical, but structurally different. Okay. And some people have better tools than others. Pre-existing conditions, including chronic infections, autoimmune diseases, cancer, and innate or acquired immunosuppression. Now, some of these might be new terms to you. An autoimmune disease is one in which your immune system attacks your own tissue for reasons we don't fully understand, okay? And that compromises your ability to fight off disease, right? Cancer is uncontrolled cell growth, and basically the cancer cell removes nutrients and oxygen from the blood at the expense of the normal tissues, and that eventually causes them to be invaded and die. Okay? And then immunosuppression is just the inability of the immune system to properly respond, and that can be something that is acquired. For instance, in people that are undergoing chemo or radiation, they're immunosuppressed, or it can be inherited. Okay, an example of that is a disease known as SCIDS, which stands for Severe Combined Immunodeficiency Syndrome. This is an inherited lack of an immune system. Okay? Your occupation is going to be a factor, right? Those that are in contact with reservoirs are more likely to come into contact with a pathogen. Okay? That includes healthcare and lab personnel, agricultural workers, hunters, prostitutes, sex workers, all at higher risk of exposure. Okay? We talk about efforts to expand civilization and the impact on emerging disease, and we'll talk about changes in climate that can alter infectious disease patterns. Okay? You can see here a map showing you global change and emerging infectious diseases. What you're looking at here is the rate of deforestation in South America, okay, and the number of cases of malaria per person. Now, why does this happen? Well, in deforestation, one of the things that you create is standing water, okay, and the standing water is breeding ground for the Anopheles mosquito, and the result then is that the vector ends up being in higher density, which means that the vector carrying the malaria is going to end up in higher density, and the result is going to be more incidence of the disease. Okay, so this is just one example. I will join everybody in the next podcast. Remember to contact me during my office hours, which are Friday from 5 to 7 by cell phone or by email if you have any questions. And I'll see you in the next podcast. Thank you very much.